In vivo magnetogenetics is a promising new approach for cell-type specific targeting and modulation of brain circuits, offering significant advantages over traditional neuromodulation techniques. This innovative method combines nanomaterials-based technology with Cre-loxP genetic engineering to selectively activate piezo-1 ion channels in specific neuronal populations using magnetic fields. Key features of this magnetogenetic approach include 1. Wireless and remote deep brain stimulation. Unlike optogenetic or wired electrode methods, magnetogenetics allows for non-invasive control of neural activity in deep brain regions. 2. Cell-type specificity. By utilizing Cre-loxP technology, researchers can target specific neuronal populations for modulation. 3. Spatiotemporal precision. The system enables precise control of neural activity in both space and time. 4. Versatility. In behavioral studies, the technique has been successfully applied to various behavioral models, including bidirectional feeding control, long-term neuromodulation for weight control in obese mice, wireless modulation of social behaviors in multiple mice simultaneously. The core of this technology is a nanomaterials-based magnetogenetic toolbox that uses magnetic nanoparticles called m-torkers. These 200 nanometers particles are composed of assembled monolayer, octahedral magnetic nanoparticles on spherical supports. When exposed to a rotating magnetic field, the m-torkers generate torque that activates the genetically encoded piezo-1 ion channels in targeted neurons. The system employs a magnetic force generator MFG apparatus, which comes in different sizes to accommodate various experimental setups, from cell-based studies to in vivo experiments with multiple animals. This magnetogenetic approach offers several advantages over existing neuromodulation technologies. 1. Wireless operation allows for studies in freely behaving animals without the constraints of implanted devices or optical fibers. 2. Long-term modulation suitable for extended studies, such as weight control in obese mice. 3. Multi-animal studies enables simultaneous modulation of multiple animals in the same physical space. The successful demonstration of this technology in various behavioral models highlights its potential as an effective and reliable research tool for neuroscience and life sciences, particularly for studies involving wireless, long-term, and freely behaving animals. Magnetogenetics and optogenetics are both powerful neuromodulation techniques, but they have some key differences in terms of precision and effectiveness. 1. Temporal precision. Optogenetics offers superior temporal precision, allowing for millisecond-scale activation of neurons. In contrast, magnetogenetics, particularly magnetothermal methods, is slower, requiring seconds to induce action potentials. 2. Spatial precision. Optogenetics can provide highly localized stimulation with precise spatial control. Magnetogenetics may offer less spatial precision due to the diffuse nature of magnetic fields, though this can vary depending on the specific approach used. 3. Depth of penetration. Magnetogenetics has a significant advantage in its ability to penetrate deep brain tissues non-invasively. Magnetic fields can pass freely through organic tissue, allowing for stimulation of deep brain regions without the need for implanted devices. Optogenetics, on the other hand, requires invasive procedures to deliver light to deeper brain areas. 4. Effectiveness. Currently, optogenetics is generally more effective and reliable. Magnetogenetics is still in earlier stages of development and, as noted, just does not work very well compared to optogenetics. It is less effective and can be frustrating to set up and execute experiments successfully. 5. Infrastructure requirements. Optogenetics requires relatively simple and inexpensive light sources. Magnetogenetics often needs more complex and expensive equipment, such as powerful magnetic field generators, which can introduce artifacts and complications in experiments. 6. Genetic targeting. Both techniques allow for cell-type specific targeting through genetic engineering approaches. This enables selective activation of specific neuronal populations. 7. Long-term studies. Magnetogenetics may have an advantage for long-term studies in freely behaving animals, as it doesn't require implanted optical fibers or tethered light sources. 8. Simultaneous multi-animal studies. The wireless nature of magnetogenetics allows for simultaneous modulation of multiple animals in the same physical space, which is more challenging with optogenetics. In summary, while magnetogenetics offers some unique advantages, particularly in non-invasive deep brain stimulation and potential for long-term studies, it currently lags behind optogenetics in terms of temporal precision, effectiveness, and ease of use. However, as the technology continues to develop, these limitations may be addressed, potentially making magnetogenetics a more competitive alternative to optogenetics. In the future, Based on the current state of magnetogenetics research, it is not yet feasible to use this technique to directly study brain function in humans. However, magnetogenetics shows promise for future applications in human neuroscience, research and potential therapeutic interventions. Here's an overview of the current limitations and future possibilities. Current limitations for human studies. 1. Early stage of development. Magnetogenetics is still in its infancy, with most studies conducted in cell cultures, animal models like C. elegans, and mice. The technology has not yet been adapted or approved for use in humans. 2. Genetic modification requirement. Magnetogenetics requires genetic modification to express magneto-sensitive proteins, or ion channels in specific cell types. This is not currently feasible or ethical in humans. 3. Safety concerns. The long-term effects of introducing magnetic nanoparticles, or genetically modified proteins into human brain tissue, are unknown and require extensive safety studies before human trials could be considered. 4. Technical challenges. While magnetogenetics offers advantages in deep tissue penetration, there are still challenges in achieving the spatial and temporal precision needed for studying complex human brain functions. Future possibilities. 1. Non-invasive neuromodulation. If developed further, magnetogenetics could potentially offer a non-invasive method for modulating specific neural circuits in humans, surpassing current limitations of techniques like transcranial magnetic stimulation (TMS). 2. Therapeutic applications. Magnetogenetics might eventually be used to treat neurological or psychiatric disorders by selectively activating or inhibiting specific neural populations. 3. Basic neuroscience research. While direct human applications are not currently possible, continued research in animal models can provide valuable insights into brain function that may be translatable to human neuroscience. 4. Combination with other techniques. Magnetogenetics could potentially be combined with neuroimaging techniques, like fMRI, to study brain function in animal models, which could inform human neuroscience research. In conclusion, while magnetogenetics is not currently applicable for studying brain function directly in humans, ongoing research and development in this field may lead to valuable applications in human neuroscience and medicine in the future. However, significant ethical, safety, and technical challenges must be overcome before any human applications can be considered. Magnetogenetics shows promising potential for treating various neurological disorders through non-invasive neuromodulation. Some key potential applications include: 1. Parkinson's disease treatment. Magnetogenetics could offer a less invasive alternative to deep brain stimulation for treating motor symptoms in Parkinson's disease patients. Research has shown that magnetic activation of neurons in the subthalamic nucleus improved motor function in mouse models of Parkinson's disease, with effects comparable to traditional deep brain stimulation. 2. Epilepsy management. Magnetogenetic approaches could potentially be used as an alternative to vagal nerve stimulation for epilepsy treatment, offering more precise control of neural populations involved in seizure activity. 3. Treatment-resistant depression. Similar to its potential use in epilepsy, magnetogenetics could provide an alternative to vagal nerve stimulation for managing treatment-resistant depression. 4. Pain control. The ability to target specific neural populations with magnetogenetics could be advantageous for developing new approaches to pain management. 5. Neuromodulation for incontinence. Magnetogenetics may offer new possibilities for treating incontinence through targeted neuromodulation. 6. Alzheimer's disease. While still in early stages, magnetogenetic techniques could potentially be applied to modulate neural circuits affected in Alzheimer's disease. 7. Expanding transcranial magnetic stimulation (TMS) applications. Magnetogenetic constructs could potentially sensitize cells to magnetic fields, allowing for the expansion of TMS applications to a broader range of neurological disorders by reducing the field strength required to elicit effects. However, it's important to note that magnetogenetics is still in its early stages of development. Significant research is needed to fully understand the mechanisms of action, optimize the techniques, and ensure safety before these applications can be translated to human clinical use. Additionally, current approaches often require genetic modification of target cells, which presents ethical and practical challenges for human applications that need to be addressed.